Well, good morning, everyone. I do not see any evidence of my slides yet on this computer, so if you could please bring them up, that would be wonderful. As I'm waiting for them, I would like to thank Steve Lippard, because it was Steve's idea to have this symposium on microbes. And I am sorry he's not able to join us this morning, but I hope that he's able to experience this remotely, and I wish him all the best. Now, when this first slide comes up, you will see a picture of a chemist, which I think is appropriate, given that Steve is a chemist and this was his idea for the symposium. Please go to the first slide. That's the second one. And this chemist is one of my favorite scientists. This is Albert Kloiver, who nearly a century ago wrote these words: "A study of microbial metabolism offers many an intriguing problem to the chemist, particularly in view of both the existing diversity and the manifest unity." So we've heard a lot about diversity so far this morning, and it's wonderful, it's remarkable. And yet to me, what's even more beautiful and even poetic is the manifest unity that underpins the metabolic diversity of all of these extraordinary microbes. Now, I find in science no greater wondrous accomplishment than the harvesting of energy, to power us and to power the earth. And we wouldn't be able to support ourselves without, within each and every one of our cells, an organelle called the mitochondrion. And this mitochondrion is where we gain our fuel, through metabolizing the food we eat, coupled to utilizing oxygen in the air we breathe to extract energy. And the oxygen in our atmosphere is absolutely vital to, our ability to live, as depicted in this beautiful picture of Neil Armstrong on the moon. Without his spacesuit, he wouldn't be able to get by because of the lack of an atmosphere on the moon. And the atmosphere that is in his spacesuit reflects the atmosphere of Earth. And in Earth's atmosphere, the most important constituent for our metabolism is oxygen. And this is so embedded into our awareness of what life needs, that it really informs our search for life on exoplanets. And so today, astronomers often use infrared planetary spectra, such as this, to look for oxygen in other celestial bodies by virtue of particular signatures. Here you see an indirect piece of evidence of oxygen with this peak of ozone on Earth, which, if we were to see this on another planet, might suggest that it's possible for life to be there. And yet, what I want to point out is that had we been using this metric to look for life on our own planet, we would have missed the first the first billion years of life's history on Earth. Okay, so we know that the Earth is roughly 4.5 billion years old. The first credible fossil evidence for the rise of microbes on the planet dates to about 3.8 billion years ago. And yet it wasn't until roughly 2.5 billion years ago that we have evidence that suggests that there was molecular oxygen present in the atmosphere. And so for certainly a few 100 million years, if not over 1 billion years, the organisms on our Earth had to invent ways of extracting energy in the absence of oxygen. Now, as has been said eloquently by our previous speakers, animals evolved in a microbial world, and they're all throughout the biosphere, in us, on us, everywhere we look. And yet, though we inhabit what we think of as a world full of oxygen, what I want to show you is if you just scratch the surface and you go down to the scales that these microbes themselves are inhabiting, very quickly we run out of oxygen. We inhabit what we call an anaerobic world. And the microbes in this world are just thriving, because if you take that phylogenetic tree, this history of life, as Victoria and Margaret have showed you previously, and you ask, what are the substrates needed to sustain all of these diverse creatures? What you recognize is that within this microbial world here, oxygen is neither required nor produced by the majority of organisms. It's really only in this little crown domain here, where the animals and plants reside, that bacteria, once upon a time, the mitochondria and the chloroplast, originated from the bacterial world and allowed, through an endosymbiotic event, the ability of animals and plants to actually traffic in oxygen. And so this is how it works for us. As I said at the beginning, our mitochondria are powering us, and they do so by taking organic carbon and effectively burning it with oxygen to generate the products of CO2 and water. Now, as any gardener knows, you have to provide water to your plants in order for them to thrive, and that's because the chloroplast is utilizing the water as an energy source that's activated with light and transforms it back into oxygen, and then it fixes this inorganic carbon, CO2, into the sugar that we're able to metabolize. And as I mentioned, the original mitochondria and chloroplast were nothing more than bacteria that figured this out many billions of years ago. But what's so glorious about bacteria particularly is their metabolic diversity. They're not restricted to this cycle that invokes oxygen. They can use so many other things. And the basis for their metabolism is really beautiful. It's unifying. And so this is what I want to teach you. It involves what we call redox chemistry, which stands for the reduction, in other words, the gaining of an electron of a compound, coupled to the oxidation, the loss of an electron from another. And so in this diagram, I have made this general, where we have one substrate, A red, which is going to be our electron donor, which will give these electrons to this other substrate, B ox, the electron acceptor. And in the process, they will be transformed. Now, a truly remarkable diversity of substrates can serve as electron donors and acceptors in bacterial metabolism, as I'll show you some examples of in just a minute. But again, getting back to the manifest unity, I want to help you see what it is about this exchange of electrons that powers cells. And it's beautiful. So in all cells where energy is conserved, including the organelles within us, our mitochondria, what is happening is that there is a series of electron transfer events that occur within a biological membrane. And it starts with the electron donor transferring electrons into this electron transport chain to a protein that is able to couple the transfer of electrons to another carrier in the membrane with the extrusion of an ion to the other side. And so this goes on until the electron is received by what's called the terminal electron acceptor in the chain. And as this electron transfer proceeds, what happens is the cell gets energized. It's like a battery. It's quite literally charged up. And it's through this wondrous charge that we are able to conserve that energy in a form that is biologically usable. Now, the variations on this theme, as others before me have mentioned, have truly shaped our world. You've already been introduced to the importance via the lovely movie Victoria just showed you of marine phytoplankton generating half of the oxygen in our atmosphere today. That's when they're using water as an electron donor to sustain photosynthesis. But a more vestigial form of photosynthesis used a dissolved form of ferrous iron, a type that emanated from the hydrothermal vents close to those that Victoria was showing you in her movie. And this is thought to have led to the deposition globally of the iron ore deposits that we mine for the iron we use today in all manufacturing. Now, in a more modern context, the ability of microbes to eat all sorts of things, including toxic compounds in petroleum like benzene and toluene, underpins the ability of ecosystems to make up for human errors and waste spills in them, to remediate them and turn these organic pollutants into benign CO2. And yet microbial metabolism isn't always doing something good, I have to confess. There are places in the world where, in the absence of oxygen, when microbe turns to other electron acceptors for respiration, this can lead to massive mobilization of toxins like arsenic, such as is occurring in Southeast Asia. But by understanding how these metabolisms work, we can begin to A, recognize them when they're happening, and B, figure out ways to control them. And so what I would like to now turn your attention to is the reality of how microbes exist anywhere, whether it's within our guts or within the sediments that Victoria studies. As she showed you, they were in aggregates. And so thank you for the question on soil. Here's an image of soil that you see on the top. These beautiful branching structures of roots mimic the branches that we see in the human pulmonary system. And yet, to me, what is the most inspiring aspect of this is that if we zoom in closely and we look at the microbes, what we find is the same theme. They're attached to each other, they're encased within tissue, or they're on the surface of a plant root, and yet when you get right down to it, the aggregates of these microbes are truly agnostic at a distance to what is beyond. So let's think about this and how we might leverage this in the laboratory to gain insight into how it is that they're able to cope when they're surrounded by so many of their contemporaries. So let's just look at a single colony developing on the surface of an agar plate, a nutrient medium where we have streaked bacteria. This is a classic type of microbiology experiment. If we take a scanning electron micrograph and look at this colony, immediately you can be impressed by how many cells are there. And I think it's even more impressive when you flip this on its side and you picture what life must be like for one of these cells in the interior. It can't be easy because we know that the rate of oxygen consumption by these organisms outpaces its diffusion into the center. And yet we also know that these organisms in the center are still very much alive. So what is their solution? What I will tell you is that they have evolved wondrous forms of molecular snorkels, which are actually very similar, regardless of the context in which you find these. All right, so the approach that I take in my lab is similar to Margaret's. I like to take the simplest possible system to approach a question that I think is a transcendent one in order to identify mechanisms that we can then use to identify what is happening out in more complex environments. And I like to focus on life in the absence of oxygen because of the simple fact that I just stated. At high cell density anywhere, oxygen consumption outpaces its diffusion. So this is truly a conserved property. And I'm inspired by Gertrude Stein's famous quote, "A rose is a rose is a rose." I substitute the word microenvironment. A microenvironment is a microenvironment is a microenvironment. And therefore, one can be agnostic in some sense about what is beyond just a few cell diameters if the question is, how are cells conserving energy in the absence of oxygen? We study this very famous organism, Pseudomonas aeruginosa. It is found in the marine environment. It's also found in soils near hospitals. It is a notorious pathogen of individuals with chronic infections. And yet, truth be told, the reason that we study it in my laboratory is because it makes these beautiful colored molecules called phenazines. And the name aeruginosa reflects the color. Aeruginosa is Latin for copper rust because of this very diagnostic color that we associate with copper when we see it on buildings that it has oxidized. And yet, if we grow this organism in what's called planktonic culture, in liquid in the lab, and we play games with it, we aerate it by introducing oxygen through this device that's called a vortex machine, which just shakes it up, what you see is that the color is toggling back and forth between this aquamarine shade, and then as it sits there, it clears. And we know why this is happening. It's because this organism is producing a beautiful pigment, which, when it is interacting with oxygen, gets oxidized and turns blue. And yet when the cells sit and they take it back up, they reduce it, they put electrons onto it, and it clears. And you can sit here all day and do this. It's really fun. For me, I knew I was never going to be cut out to be a marine biologist because I get very seasick on any boat I go on. And so this is a better place for me to get my thrills with microbes than being out in the ocean, despite the phenomenal things that we saw Victoria show you. And yet, what we learn from doing this in the laboratory actually informs our ability to appreciate what's going on in the bottom of the ocean. And here's why. So we had a hypothesis, which was that the production of these colorful pigments might actually allow the cells within the interior of these films to remain metabolically active. And one of the tools we use in my laboratory is that of genetics. We try to understand how things work by breaking them and seeing the effects. And so we figuratively broke the ability of these organisms to produce these lovely pigments by generating a mutant strain that was unable to synthesize them. We call this mutant delta fuzz. And then on the other hand, we generated a different strain which overproduced these molecules. And in the middle, we looked at the normal strain, which is also called the wild type. And what you're seeing now are colony biofilms forming on the surface of agar that are developing in size over the span of a week. And you can see by day 7 there is an extraordinary difference. The one on the right, which is overproducing these molecules, is able to make a lovely, thick biofilm, and it's nice and smooth. Whereas on the left, the one that is unable to produce these pigments has many wrinkles, and it's colonized a much larger surface area. You can make the analogy to how, for instance, in Los Angeles or New York, we solve the problem of breathing room. In New York, there's skyscrapers. In Los Angeles, we spread out. Similarly, that's what these cells are doing here. In the absence of having these molecules that they can use to sustain life in the interior, they are restricted to inhabiting only areas within this colony where they can access oxygen. So let's take a deeper look and connect this back to the rules of metabolism that I taught you at the beginning. If we follow the growth of a single cell in culture over time, what you will find is that as it grows, oxygen is depleted. When the cells are multiplying, they're utilizing this oxygen. They keep exponentially growing. And then finally, at this point when they're literally running out of oxygen, this is when they time the production of these pigments. Then they stop making them and start recycling them. It's an extraordinary sustainable solution. Because in effect, what these organisms are doing is producing a self-made electron acceptor in order to conserve energy. And the reason it works so well is that there is something at a distance that's able to recycle them. And this process of recycling is achieved through what we call extracellular electron transfer, very similar to what Victoria alluded to, that allows these methane-oxidizing archaea to couple to sulfate-reducing bacteria. Only the molecular strategy is a little bit different. So in this system, it sustains life within these colony biofilms in the core. But importantly, what we've been able to show is that this strategy, as I said in the beginning, is really agnostic to context at a distance. It's only the microenvironment that matters. So this biofilm can be on a host tissue. It can be on an electrode, where instead of having oxygen accept those electrons, an electrode takes them and then generates current. Or it could even be on the surface of a mineral. The cell in the middle doesn't know, and I would argue doesn't really care. And yet what we can do is make use of the versatility of this system to really further probe and understand the strategy whereby this is successful. And so I'll just end by showing you a little bit of data from a recent collaboration I had with our chair, Jackie Barton. And so what we've done in this system is develop biofilms growing on an interdigitated electrode array that has these 2 electrodes on the edges. This is the zoomed in part of this little device here, which we drop into a suspension that has our bacteria growing, producing this pigment, and there's no oxygen therein. And then we allow a biofilm to form, and we take a look at it. So here are those 2 electrodes. what you can immediately see, it looks like a work of modern art. In red, we have the biomass, and in blue, interestingly, we have a lot of extracellular DNA. And we can ask, is it that these pigments I've been saying are critical to sustaining these biofilms? Is that really the case? By making use of the fact that we can measure current when they react with the electrode. And so here we're looking at current over time, and here we're looking at this slow, steady heartbeat of metabolism, if you will, for the wild-type organism. We are unable to see any current being generated in the mutant that doesn't make these pigments, and yet when we add back these pigments, it restores the heartbeat of life. And so the answer in this particular case to how it is that these biofilms in the interior are able to sustain a slow and steady pace of life through a molecular snorkel, if you will, is that the interaction of these pigments with DNA allows them to be retained in such a way that promotes efficient extracellular electron transfer. And yet, I have only told you the story of one organism and one class of pigments. We can go out into the soil and we can find many other bacteria that produce different colored pigments that have different biological functions. We can make art like the Munch Scream, but more importantly, what we can do is be inspired by the unity and the diversity of microbial metabolism, and through studying how it works, gain insights that really wind up actually translating very directly to solving important problems across many realms of human endeavor. So with that, I'll say thank you, and we can go right into the Q&A session.
Jim Peebles, Princeton, New Jersey. The mention of the origin of life and the bucket of seawater that contains so many microbes reminds me of the speculation that also in that bucket are many fragments of DNA from all kinds of critters. And the question would be if, when life began, there were experiments along different lines, so that there were other methods of genetic transformation of information, there might be molecules in that water left from other forms of life. Does that hold any water?
That is just a wonderful question, and I can't give you a yes or no answer, but I can tell you one of the things that we've been looking at with environmental samples are gene sequences that are coming from the viral world, or what we think are the viral world. So this is capturing really very small particles out of the environment. And I'll give you an example just from the methane seep environment that we looked at alone. When we sequenced these very small particles, we recovered 80% totally new to science diversity of things, some that look like viruses, others that don't, and we can't make sense of it right now. We just know there's a lot of novelty there, and we know there's a lot of these small particles that have DNA because we can stain them. And so this is, I think there's probably a history that's hidden in the world of viruses that might give us more insight into what was going on. I don't have any reason to be able to say for certain, but I think that viruses probably evolved very early on in the game, and so have been co-evolving with all sorts of life. And so I think there might be some secrets that we can learn by looking at them. Yeah, great question.
Say your name. This is Ruzhna Baichi. I am a systems engineer, roboticist, so I come from that. And I was caught by the title, the diversity. Pardon me? Yeah.
A little louder, please.
Say it again. What?
Just a little louder, if you could.
Okay. Sorry. So I'm caught by this the powerful unity and diversity. So where is the unity and where is the diversity? Is the diversity, in my business we talk about input, output, and the process in between, the structure in between. So where is the unity and where is the diversity?
So that was one thing I was trying to illustrate with the cartoons in my talk demonstrating how energy conservation works. The unity is that there is a generalizable mechanism that powers all cells with regard to coupling electron transfer to the generation of a gradient that can be harnessed to drive work.
So,
Now,
excuse me, let me just say, so what scale are you?
I'm talking about at the scale of a single cell or an organelle of a membrane,
Okay.
the biochemical. So
you
the
are
biochemical unity
on the
Yeah.
cellular level.
on the cellular level.
And not on the systems level.
I'm talking on the cellular level very much.
Got it.
Yes.
Got it. Okay, thank you.
It's a great question. 'Cause
Question in the back.
we don't understand microbial community assembly and what are the rules there, and are there some overarching principles? This is an active area of research. So.
Yeah.
Great.
I think we'll go to we can't hear you, so maybe we'll go to the...
I have a very short question. Joanne Eisenberg, Boston. My question is, with everything we learn every day about viruses, I'm still surprised they're not considered life. Are we going to upgrade viruses to that level, or we are still considering, for some strange reasons, that they're not?
You want to take that, Margaret? Do you want
Margaret, do you want to take that one?
Well,
Yeah.
I think that most of the time they are dependent upon cells of either bacteria or human cells. And so the idea is, and oftentimes you can see that they're derivatives of those cells. I mean, it's actually quite controversial. And when you were saying this, I recall I went to a meeting at the Sorbonne years ago, and there was a person who stood up and talked about this very question. And he provided a very convincing presentation that they are indeed life. And so when you asked the question, I thought, I don't know.
Yeah.
Can I add something?
Yes, please.
Wonderful question, thank you. So, in answering that, my mind can go to a colleague, an anthropologist. An American anthropologist. Her name is Elizabeth Povinelli. In one of her books entitled Geontologies, she discussed three figures, and one is the virus, because what is called in our discipline biopolitics, so is the power over death and life. So if you think back at the king, right? The king was this powerful person that could decide who stay alive and who have to die. And Povinelli say, biopolitics is not only that. It's not just the power over death and life. More profoundly, at the core of biopolitics, that there is the power to decide what is life and what is not life, right? So, and this speaks a lot to these things, and the virus she takes the virus as the figure of that little thing that blurs this difference between life and non-life. So certainly there are scientific reasons, but somehow there are also cultural and political reasons why we have this difficulty of accepting the virus also as a life form. As we have difficulties to accept rocks or the planet Earth as a living thing. So, yeah, this is the anthropologist's reply.
Can I add one additional thing? Yeah, so the other component of this is, again, by being able to sequence very deeply into the environment. So we've discovered these giant viruses, and it used to be that viruses were not known to be able to do any of their own replication, yet we find some of the translational machinery is encoded in these giant viruses, and they can be infected by other viruses, right? So the lines are definitely blurring in our concepts of what is living and what isn't. I like to think that the jury is still out, and I'm definitely not going to be dogmatic about it. When we think about microbial life in the oceans, there are 10 to the 23rd viral infections that are happening every second. And it's not like they get infected and they die immediately. Those viruses are manipulating the metabolism of those cells. And so the field has turned to starting to call them viro cells, because they don't have a metabolism now of the host. They're sort of been hijacked by these viruses, and we don't really have a good handle on how that is impacting the overall processing of matter on the planet.
Can I say one more thing, too? In health, one of the interesting things was when we began to understand the microbiome. On the microbiome, we largely focused on the bacteria that are associated with the human body and animal bodies and so on. But there's also what we now know is a virome. In other words, there is a set of viruses that actually confer health. And so to give an example is the herpes virus. When you have an outbreak of herpes, it's not a good thing, of course. But the latent carriage of herpes confers heightened resistance to getting certain types of bacterial infections. And so and this is being discovered again and again and again, and your gut microbiota is shaped by the phages that are in your gut. Isn't that right, you guys, the bacteriologists? Yeah, but I mean, there's the viruses are, and fungi as well. So an example of the fungi is Candida albicans. And Candida albicans has always been considered a pathogen, always. But every single person carries Candida albicans. And it only is a pathogen when you're immune compromised or something like that. And so the question is, for health, do you require Candida albicans? Likely so, in my opinion.
Other questions? Yes.
I have a question. Betty Ruth Walter, University of Pennsylvania, Philadelphia. You all know so much about what is in places of the earth, such as the ocean, which is my question. It concerns the ocean and man's relationship with the ocean. For example, I live by the sea in the summer. There is a huge discussion about these windmills. Putting them in the ocean, is it going to better our lives? Are we going to have all kinds of free energy? Or are we ruining our lives by what we're stirring up at the bottom of the ocean? Or what is in these microbes? Is there anything that we know, that you all know, that can help us make these decisions?
Wow, that's a great question. I mean, in general, I think wind energy is a good idea. I have not heard whether or not putting these wind turbines in the ocean, if it actually physically is mixing the ocean. But I would say that would not be necessarily a great thing, depending on where that's happening. There are natural places where we have upwelling that has to do with the topography of the seabed, and that is actually a critical way of getting nutrients into the surface water that supports all of those photosynthetic organisms. And so there is some benefit of bringing deep, nutrient-rich, deep water back to the surface, but that goes only so far, right? So it sort of depends strategically. If that really is promoting mixing, I would say we'd have to think very deeply about where we put these things, not just for convenience and out of sight of the homes on the coast, but also in terms of how it's impacting the ecosystem.
Yes.
If I can add quickly, I think we are really in this scientific transition in which we are moving from considering the environment as just made of resources, and resources go back to this idea of somehow scientific certainty about what these things are. And the microbiome, among other scientific discoveries, are making us aware that before to intervene, we really have to know the complexity, and we still have a lot to learn to understand the complexity, because otherwise, like, we go back to my example, the antibiotic resistance, then we might create more problems then.
Next question over there, yes.
First, a question about the sociology of science and then about science. I note with admiration that 4 of the 5 people on the stage are women from Caltech. Does that reflect a monopoly on this subject by Caltech, or is that a network effect? How does one account for this statistically improbable observation?
Am I supposed to answer that one?
And before you answer, let me ask my scientific question.
Oh, good.
Please. We know from recent experience that human exposure to viruses from habitats with which humans are not familiar can cause worldwide infectious disease pandemics. For example, possible origin of the COVID virus, SARS-CoV-2. And I understand that NASA has protocols for checking the microbiological activity of samples brought back from space to see whether there's any risk of infection on this planet. We are bringing up samples from the deep ocean in habitats that we have never encountered before. Is there a governance structure? Is there a scientific review procedure? How do we know what risks we are putting ourselves exposed to through these explorations, and what are we doing about it?
Yeah. To answer your question, there aren't currently any protocols in place when we collect samples from the deep ocean. There are definitely protocols on resource extraction from other countries. So we take it, we recognize that the genetic information that is existing across the globe is a valuable resource and can be used for biotechnology. And so now there's some agreements in place that those resources will go back to the country. But that's a different question than, are we potentially going to be pulling something up that could be dangerous for us? What I would say about a deep ocean environment is it's very different than our bodies. And so it would be very hard for anything that lives in the deep, dark, cold, pressurized environment to actually get into our system and do any havoc. I can only tell you in the 30 plus years I've been doing it, I'm still here, so there's that. But there are examples of like the thawing Siberian permafrost where they have actually been able to recover giant viruses and get them to infect amoeba living today, right? So that's also, from an evolutionary standpoint, it's fascinating, right? Something that was living, maybe I think several thousand years ago now is still capable of infecting viruses today. So it's a concern. I might think about it more now that you've said this,
It is, yeah, exactly.
Yeah, yeah.
It's not
Right.
implausible at all, and I don't think, to be very fair, I just don't think people have really looked at this, quite frankly. And interestingly, maybe it's because there's a knee-jerk assumption that it's so very different, and yet when you get down to some of the details, it really might not be. However, there are regulations, certainly for the transfer of soils around the country and between countries. We all know this when we go on the airplane, you have to sign the attestation that you're not bringing in fruit or something from another country into the United States. That's highly regulated, and yet we haven't made those regulations for deep ocean sediments.
And what is also highly regulated are research that also in the outer space. So there are strict protocols to allow that there is no infection, not only of material collected in outer space that has the risk to infect humans and planet Earth, but also that we can infect and bring new life into other planets.
And I think also that there are things about the human body, like the temperature, and also, you know, microorganisms, and as well as all organisms, are shaped by the temperature and pressure at which they occur. And the molecules don't work like deep sea animals and deep sea bacteria. When you pull them up, they yeah. And, you know, and temperature is really a strong selection.
I think there's a question on the balcony.
Yeah, hi, Mike Hout from Brooklyn, New York. My question has to do with history rather than the deep sea difference. Is our microbiome more diverse or less than Benjamin Franklin's?
Ooh, good question.
There's a lot of work that's being done now comparing the microbiomes of cultures that live close to the Earth, like the Yanomamo and people like that, and the people in Malawi, and they have incredibly diverse microbiomes. And also, there has been a correlation made by the biomedical community that the more diverse the microbiome, the healthier the microbiome. And we are losing, in the first world nations particularly, we are losing the diversity of our microbiome. A really good example is Helicobacter pylori, which, as many of you know, is the causative, will cause ulcers and gastric cancer in post-reproductive individuals. It's been found by Martin Blaser that he's a physician, scientist. It's been found that Helicobacter pylori is actually really critical for young people because asthma and autoimmune disease and certain things like that are helped by carriage. You know, the carriage of Helicobacter in young people is a really good thing, it turns out. Well, Helicobacter, we have lost, at the turn of the 20th century, 90% of the Americans carried Helicobacter, now 10%.
Wow. Yeah. And with the technology at hand, microbiome scientists are also able to study the microbiome, for example, of mummies. A very good collaborator of mine, Nicola Segata, and his team, for example, have collected the microbiome of Ötzi, which is this mummy living in the glaciers between Italy and Austria. So, yeah. They reconstructed the last lunch or dinner before dying of these mummies, so they were able to see through the micros, like traces, not really the micros and traces.
Other questions or comments? If not, thank you all, and let's thank the panel once again for a great morning.